Okay, today we are going to discuss about what is pathogenesis, what is the role of enzymes, toxins, growth regulators, and polysaccharides. And before that, we already discussed about different kinds of like way or medium, we can say that vector by which the pathogen disperses the particular disease from one place to another place, and how the pathogen infect one plant to another plant. Okay, so today, what is the pathogenesis? Pathogenesis, that means from the starting or from the entry of the pathogen, what is the procedure and what is the process by which the pathogen can cause the disease in the plant. So that is called the pathogenesis. Okay. So first is we are going to talk like role of enzymes. So there is the main components of cellulose are pactic substances like cellulose, hemicellulose, lignin, and a small quantity of protein. Okay, you know about that thing. These are the components of cellulose by which the cellulose is made up of. So there is the composition of cell wall which divided into three regions. Three regions like middle lamella, primary wall and secondary cell wall. Okay. So first is the primary lamella which is made up of pectin. Pectin, that is a component of cell wall. Okay. So middle lamella, which is made up of pectins, that is intercellular cement which binds the cells together. Okay, intercellular, that means between two or more cells. Okay, not only in a single cell. Then primary wall, cellulose, peptic substances, and secondary cell wall, which is entirely cellulose. Okay, so these all are considered like cell wall, which is made up by like cellulose, hemicellulose, lignin, pectin, etc. So the epidermis of plant is covered by cuticle, whose major chemical substance is cutin, in addition to cuticular wax. So the epidermis of plant which is covered by the cuticle, so that is a hard. Okay, so sometimes it is very difficult to penetrate their cell wall for the pathogen. That's why here we are going to talk about what is the role of enzyme, what is the role of toxins, and how the pathogen which will be like disappear or lyses the cell wall. So first is the cuticular wax. Cuticular waxes are made up of long chain molecules of paraffin, hydrocarbons, alcohol, ketones, and acids. So, most of the fungi and parasitic higher plants which penetrate the wax layer by means of mechanical force alone. So plant waxes are found as granular or rod-like projection or as a continuous layer outside within the cuticle. So there is the wax formation is a continuous process, and it is not a terminal phase in the development of the leaf. After that, cutin Cutin, it is an incellular polyester of unbranched derivatives of C16 like carbon 16 and carbon 18 hydroxy fatty acid. Cutin is admixed with waxes or upper side and with pectin and cellulose on the lower side. Then the cutinase, that is break cutin molecules and release the monomers. So, like the enzyme which lyses the cutin, that is cutinase. Always there is the ase will be suffix of the enzymes. Okay, like cellulose, which is degraded by the cellulase. Then pectic substances, there is the major components of medulla, intercellular cement that hold in place in the cells of plant tissue. Okay, so pectic substances are of three types, namely pectic acid, non-methylated units, pectinic acid, that is 75% methylated, that means there will be the addition of CH3 group, that is galacturonan units, and pectin is 75% methylated units. So the pectic degrading enzymes, that is pectinase or pectolytic enzymes, pectolytic enzymes, these are the three types: pectin methyl esterase. In short form, we can say that PMEs breaks ester bonds and remove methyl group from pectin. So there is the polygalacturonase, that is split pectin chain by adding a molecule of water and breaks the linkage between the galacturonan units. Then pectin lyase split pectin chain by removing a molecule of water from the linkage. And whenever the bonds will be break, only in that case the pathogen which can enter inside the plant. Okay, that's why whenever there is the attachment of the pathogen or the host, then first of all there will be the role of the different types of enzyme, like by which component the cell wall is made up of only. So the same enzyme, they will be degrade the cell wall, and after degradation of cell wall, the pathogen, those can enter inside the plant. Okay, and then start their multiplication. That's why enzymes have a very important role. After that, cellulose. Cellulose is a polysaccharide which made up of chains of beta D glucopyranose units, and that is the cellulase. Like degradation of cellulose by cellulase. Okay, that is the enzyme which used for the degradation of cellulose. So the cellulase is a degrading enzyme play a role in softening and degradation of cell wall material and facilitate easy penetration and spread of pathogen in the host. If there will be no breakdown of the cell wall components, then pathogen can't enter inside the plants. So these are the major consequences of primary cell wall, that is hemicellulase. Hemicellulase, which degrades the hemicellulose lipids, various type of lipids occur in all plant cells. Then lipase enzyme, which will be degrade the lipids, lipolytic enzymes like phospholipase, glycolipase, which hydrolyze the lipids and release the fatty acid. Fatty acids are the units of lipids. Okay, like what is the unit of protein? Tell me, what is the unit of protein? अरे बताओ तो सही, किससे मिलके बनता है प्रोटीन? अमाइनो एसिड। तो फिर बोलते क्यों नहीं हो? So same thing here, like the fatty acids are units of lipid. Then starch is the main reserve polysaccharide found in plant cell. Polysaccharide, that means that is a carbohydrate. Okay, and that is a polysaccharide because there are more than 10 monomers. Then amylase, which is a starch degrading enzyme. Lignin is found in the middle lamella as well as in the secondary cell wall of xylem vessels and the fibers that strengthen plant. After that, toxins, and what is the role of toxins? So first is toxin, that is microbial metabolite directly toxic to the cells of the suspect, that is the host. Okay, so they will be excreted exotoxin or released by the lysed cell, that is endotoxin. Difference is there. Here two words are there. One is the excreted, another is the released. Okay, the meaning will be same, but excretion, that means these will be exotoxin or these will be outside of the plant. Okay, so for outsiding we are using here excreted word, and released by lysed cells, that means these are the endotoxin and these are released inside the cells. Is that clear? Excreted, that means exotoxin which will be excreted outside the plant cell, and endotoxin, those released by the lysed cells. So there is the different from enzymes, like they do not attack structural integrity of host tissues and affect the metabolism of the host. So how are they differ? Toxins act as a protoplast of the cell. And enzymes act as on structural integrity. Structural integrity, that means, like, first off, there will be lysis of the plant cell wall. So cell wall, that is a structural components. Okay, that's why here we are saying that the enzymes act as on structural integrity, and toxins, which will be act as a protoplast of the cells. Then the toxic hypothesis, that is the Luque and Wheeler in 1955. A toxin should produce all symptoms, that is characteristics of the disease. Sensitivity to toxin will be correlated with susceptibility to pathogen, like sensitivity equals to susceptibility. Okay, that means it will be affect earlier. So for that we are using the sensitivity or susceptibility. So, toxin production by the pathogen will be directly related to its ability to cause the diseases. Okay, then exception is there, like Victorin, the toxic metabolite of Cocciolobus victori. The vast majority of plant diseases associated with this toxin fail to exhibit all the above characters. After that, classification of toxins, that is by Wheeler and Luque in 1963. That is two types, like according to the source of origin, three broad classes, namely, first is patho, that is toxin, second is the vivotoxin, and third type is phytotoxin. Okay, so here, if the any pathogen, sorry, any toxin which is related to the patho, so patho, that means disease. Okay, so the pathotoxin, those play a major role in disease production, then produce all or most of the symptom characteristics of the disease in susceptible plant. Then our second type is vivotoxin. Vivotoxin, like substances produced in the host plant due to host pathogen interaction. Okay? So whenever there will be interaction by the pathogen with the host, then this type of toxin will be called vivotoxins. Then the causal role in disease is merely suspected. It is not established. These are the products of parasites which induce few or none of the symptoms caused by the living pathogens. So they are non-specific, and there is no relationship between toxin production and pathogenicity of disease causing agents. And then third type is phytotoxin. Phytotoxin, these are the substances which produced in the infected host by the pathogen or host. So the substances functions is the production of the disease. Phyto, that means which is related to the plants. Okay. So these substances function in the production of the disease, but is not itself the initiating agent of the disease. So that will be called phytotoxins. After that, classification which based on specificity of toxins. So first is host specific and another is non-host specific. So host specific is there, that means selectively toxic only to susceptible host of the pathogen. Non-host specific, that is no host specified and can also affect the physiology of those plants that are normally not infected by the pathogen. That means if toxins are host specific, then they will be toxic only for the susceptible host of the pathogen. Like the host which will be susceptible for the pathogen and only they will be cause the disease in the particular plant. But if non-host specific is there, then only the toxin, those affected the physiology of the plants, so that are normally not infected by the pathogen. Then the primary determinants of disease and the non-host specific are secondary determinants of disease. That means if host specific toxins are there, then we can easily recognize the disease. Okay, but if non-host specific is there, that means we can't recognize as soon as and as comparison to host specific. Then the host specific, the host specific which produce all the essential symptoms of the disease, and the non-host specific, so they will be produce few or none of the symptoms of the disease. Then host specific, for example like Victorin or T toxin, and the non-host specific, that is 10 toxin or tab toxin. After that, effect of toxins on host tissue. So effect of toxins on host tissue, in which like there is the changes in cell permeability. Toxins kill plant cell by altering the permeability of plasma membrane, so thus permitting loss of water and electrolytes, and also unrestricted entry of substances including the toxin cellular transport system, especially hydrogen ion or potassium ion exchange at the cell membrane is infected. Then there is the disruption of normal metabolite process Those increase in respiration due to disrupt salt balance, malfunctioning of enzyme system. For example, like pellicularian inhibits the polyphenol oxidase upcoming of oxidative phosphorylation. So there is the interfere with the growth regulatory system of host plant, for example, like restrict development of roots. Those are induced by fusarium. After that, growth regulators. So growth regulators are two types, like growth promoting or growth inhibiting. Promoting, that means those will be increase the growth of the plant. Okay, so these are the growth promoting, and inhibiting, that means those will be suppress the growth of the plant. Okay, so growth promoting, in which the auxin, gibberellin and cytokine. So these are the growth regulators. Those increase the growth of the plant. That is like auxin, which will be increase the growth of the branches, gibberellin and cytokines. Growth inhibiting substances, that is dormin, ethylene and abscisic acid. Okay. So they will be inhibit the growth of the plants. So the imbalance in growth promoting and growth inhibiting substances, which causes the hypertrophy, excessive increase in cell size, and atrophy is decrease in cell size. Hypertrophy, that means due to the increase in the cell size, we can say that there is the imbalance. Okay, so that will call the hypertrophy, and atrophy, that means those decrease the cell size. Symptoms may appear as tumor, galls, with those are known, which is broom, stunning, excessive root branching, defoliation, and suppression of bud growth. Then the polysaccharides, like fungi, bacteria, and nematodes, those release varying amount of mucilaginous substances that coat their bodies and provide the interference between the outer surface of the microorganism and its environment. So there is the slimy polysaccharides is of utmost importance in wilt disease. Polysaccharide, like fungi, bacteria and nematodes, those release varying amount of mucilaginous substances that coat their bodies and providing interference between the outer surface of the microorganism and its the environment. So the role of slimy polysaccharide is of utmost importance in myself Manju Sharma from Suresh Kandiya University, Faculty of Agriculture, and the subject is Fundamentals of Plant Pathology. Today we are going to discuss about how the disease which is spread from one place to another place or from one plant to another plant. So firstly, let's come to the differentiate or difference between seed infection and so first is seed infection. Seed infection for some time and established its relationship with the seed tissues. Example is loose smet of wheat, where the fungus grows in the embryonic tissues and becomes dormant when the seed enters dormancy. So that will be called seed infection. Seed infection, we can say that the seed is infected at the stage of embryo or at the embryonic stage. Seed infection when the fungus of the pathogen is present on the seed coat. That means whenever the pathogen which will affect only on the upper surface or seed coat of the seed, then it will be called seed infestation. So seed is infested and act as the transport of the pathogen. Then soil as a mean of autonomous dispersal, soil-borne facultative saprophytes or facultative parasites which survive through the soil. That means if the pathogen is a soil-borne pathogen or if it is facultative saprophyte. Saprophyte, we can see that like facultative, those have alternate host. If their host is not present and if the host is dead, then the pathogen which can also survive or feed on the other material. So here is the dispersal may be by two ways. First is the movement of pathogen in the soil or by its growth in soil. That is dispersal in soil. That means when the soil is dispersed and when the pathogen which will be dispersed in the soil. The movement of the soil, that is dispersal by the soil. So two things are there. Like first is dispersal in soil and another is dispersal by soil. So first is dispersal in soil. Three stages. First is contamination of soil, which takes place by gradual spread of the pathogen from an infested area to a new area. That means uh, it will be take place by gradual spread of the pathogen from an infested area to a new area. Second is growth and spread of a pathogen in soil. So once the pathogen reaches soil, it can grow and spread based on its ability to multiply and spread. Another is adaptability to soil is governed by high growth rate, rapid spore germination, activity, capability to produce antibiotics, and tolerance to antibiotics which produced by other soil microorganisms. That means whenever high growth rate, rapid spore germination, better enzymatic activity, capability to produce antibiotics, and tolerance to antibiotics produced by other soil microorganisms. So, on the basis of this competitive saprophytic ability, the pathogen in soil can be of three types. One is specialized parasite. Second type is unspecialized facultative parasite, which can pass their entire life in the soil. That is Phytium species and Phytophthora species. And the third type is soil-borne obligate parasite. So let's come to the specialized facultative parasites. So the saprophytes can pass their life in soil in the absence of host plants. That means if the host plant is not there, in that case the pathogen will be, and the pathogen those can pass their life in the soil, but they depend more on the residues of the host plants are there. In that case, this kind of parasites easily survive. For example, Armillaria melia, Ophioglossus, Daminates. Another type is unspecialized facultative parasites can pass their entire life in the soil. For example, Phytotoxin species and Phytophthora species. Third type is soil-borne obligate parasite such as Plasmodiophora, Brassica, and another is Synchytridium endobioticum, which require the presence of active host. And the third is persistence of the pathogen in soil. So the pathogen which persists in the soil as dormant structure like oospore, chlamydospore, smut-spore or sclerotia. So first is like oospore, in which the Phytium, Phytophthora, Sclerospora, Chlamydospore, that is Fusarium, Smut-spore, that is Astillego, and Sclerotia, that is Rhizoctonia sclerotium. Then second thing is dispersal by the soil. So the pathogen which dispersed by soil during cultural operations, in which the first is agricultural implements, second thing is irrigation water, third is workers' feet, etc. By the agricultural implements, like we are just using these things. In that case, the pathogen which will be dispersed by the soil, by the irrigation water, and by the workers' feet. The transfer of soil from one place to another, along with propagating material, is the most important method of dispersal of pathogen. So by this, the transfer of the soil from one place to another place, and it will be along with propagating material, is the most important method of, dispo- of dispersal of pathogen. That is transfer of various new planting the seedling. And similarly, parts of fruit trees is transported with soil around their roots can transmit pathogen present in the nest to the occupants. Third is that plant and plant toxins as a means of autonomous dispersal. One part has been seen for field reduce and plant accumulates of potato was introduced not from